A very good afternoon to one and all present here. I, Dr. Ramifications, Assistant Professor from the Department of Biotechnology. I would take an immense pleasure to welcome you all to the 5th day of one-week professional development program on avenues of biotechnology. This professional development program aims to enrich the knowledge of the faculties through interaction with eminent personalities from industries and academics. The knowledge sharing will improve the learning process for the faculties and benefit the student society. Now, I would like to invite my colleague, Dr. Vinod Kumar, Assistant Professor from the Department of Biotechnology, to introduce our today's resource person. Sir?
Thank you, ma'am. Good afternoon, everyone. Good afternoon, Dr. Arvind, sir. Very thankful to you for accepting our invitation in a very short notice. Thank you again, sir. And it's time for me to introduce today's research person. He is very experienced academician and a researcher. And he's Dr. J. Arvind. He did his BSc in Biotechnology at GRD College from Coimbatore and MSc in Environmental Science at Bardiyar University. Then he did in MTech in Environmental Engineering at Vellore Institute of Technology and Vellore, and he did PhD in Biotechnology at Vellore Institute of Technology. He have wealth of experience in teaching as well as in research, and he have more than 15 years of teaching experience. As soon as he completed his PhD at Vellore Institute of Technology, he joined there as assistant professor, and he continued to work there for few more years before joining in Kumaraguru College of Technology, Coimbatore. In total, he worked as assistant professor for almost 9 years. Then he moved to Ethiopia, College of Chemical and Biological Engineering, Addis Ababa Science and Technology University in Ethiopia, and he joined there as a professor in 2016. Later, he moved to GRD institution in Coimbatore. Currently, he is working as an adjunct professor at Department of Biotechnology, Savetha School of Engineering, Savetha Institute of Medical and Technical Sciences, Chennai. And his research credentials are very commendable. He has published more than 75 research papers in very reputable journals, and the highest impact factor of his own publication is around 15 impact factor, and the total cumulative impact factor lies around more than 100. And the total citation, his articles and review papers received are more than 1,100 citations, which suggests that his research credentials are very commendable in nature. In addition to publishing research articles and reviews, he also edited several volumes of books which have several thousands of downloads from the internet source, as well as selling of several hard copy books. And he has contributed by publishing several chapters in edited volumes by other researchers worldwide. In addition to that, he also involved in editorial activities, such as he is acting as an editorial board member as an associate editor in International Journal of Environmental Science and Technology. And in addition to that, as a reviewer of manuscript, he has reviewed manuscript of more than 500 manuscript in around 25 different journals published by Springer as well as the Elsevier research publishers. Apart from that, he also is a regular invited speaker. He also has chaired several sessions, and he also acted as a resource person in several international conferences, workshops, and seminars. So we are very happy to invite such an experienced person to speak in our professional development program. Finally, we welcome you, sir. Thank you so much for accepting the offer again. Over to you, ma'am.
Now, without further delay, we will turn the time over to our speaker, Dr. J. Arvind, to deliver his lecture on sustainable approach for handling emerging contaminants through biotechnology. Sir, over to you.
Good afternoon.
Yeah, good afternoon, sir.
My screen is visible to all.
We can able to see you, sir, but we are not able to see your screen.
Yeah.
We can do your screen share, sir.
I did just a second. Is it visible now?
Yes, sir. Yes, sir.
Yes, sir.
Yeah.
Yes, sir.
Sorry for the delay.
No issues, sir. Please.
Very good afternoon to one and all. It's my pleasure talking to you. I hope that you had a fruitful 4 days with very interesting topics, broad topics, and some very specific narrow down research topics, all these things. Today, I will be speaking on sustainable approaches for handling emerging contaminants through biotechnology. This is actually a very broad topic where each section can be talked in depth. Collectively, when I was approached by a professor, I thought of taking a topic, maybe a specialist topic, but the necessity of the particular PDP where a talk should be interesting to the participants. We don't know about, it's a bit tricky to give a lecture on online rather than live audience in person. So I finally decided on this topic. And the two important reasons for this topic selection is that you can see that in biotechnology, especially in environmental biotechnology domain, the current research is focused on emerging contaminants. And when you talk about any research, whether it's academia or industrial level, you take any innovation, we always think about the scientists and other eminent persons talk about sustainable approaches or sustainability. So this is a very need of the hour. So that's the reason for me to give you a talk on this particular topic. Before we go into, I'm not going to give you a very in-depth this thing. I'm going to just talk on the general aspect, then give you different pointers so that there will be so many openings for people who have very good research instead to start their own branch of research in this particular topic, whether it's bacterial or microbial or algal or any hybrid system where you can go for mitigation of this emerging contaminants. So I've given you a lot of openings. So that it will be interesting talk on how to proceed on using biotechnology techniques to do the handling of this emerging contaminants. So we'll start with this, what is sustainability? Sustainability in a nutshell is we need to use our resources in such a way wherever applicable possible, so that you don't actually compromise the need of the future generation. So that is the definition given by the United Nations World Commission of Development and Environment. So this is very important, and people may would have known about sustainability and the component of sustainability, but we need to always remember that sustainability always includes three elements and the harmony between these three elements: economy, society, and environment. Whatever endeavors we do as a researcher or industry, most of the research will be focused on the economical outcome, or it can be socio-economical outcome. Very rarely we go for environment or focus all the three elements coming into harmony. So that is what sustainable brings to the forum. So what is the current situation now is we have something called a very unsustainable environment, unsustainable situation, and moving towards sustainability or sustainable environment or sustainable society, we need to go through a narrow corridor of sustainable society. At present, we have ever increasing population. There is a very high increasing, ever increasing demand for resources to cater to the needs of the increasing population. And simultaneously, we have a decline in resources, availability of land space, agricultural resources, ecosystems. And one more component is that to add misery to the situation, the increasing demand and the declining resources have put forth tremendous pressure on research activities and whatever product industry is focusing, they are trying to balance. Unfortunately, without sustainability, you have a problem of pollution, where it may give rise to harm to the future generation or the future environmental situation. So we need to go for some sustainable innovative solutions, some creative solutions where it can lead to sustainable approaches, so that the sustainability is maintained. So going to the next component of the topic, what is emerging contaminants? So the interesting feature of this emerging contaminants is what emerging contaminant today may not be tomorrow. So it keeps changing. So that is the beauty of these emerging contaminants. So as you can see the definition, so it can be either naturally or synthetic origin, it can be chemical based, it can be microorganism, it can be anything. But the main aspect of emerging contaminants is it is in focus. It has some potential to cause harm to the environment in the future. So that is to give you a simple terminology, what is emerging contaminants. So all those components, all those elements, all those pollutant, whatever it may be, which fit into this definition can be grouped under emerging contaminants. So when you want to discuss about the emerging contamination terminology, the previous slide split up. So either it can be synthetic or natural, it can be either a biological matter or a chemical substance. It is generally not routinely monitored because in the past or in the present, it is not a threat. It's not a contaminant. It's not a problem causing or environmental problem causing substance, but it may lead to future environmental hazards, environmental pollution. So that is why it's called as emerging. So we need to have some regulations, periodically check and change the classification, change the components of the emerging contaminants. So this is done by the US EPA and with support with inputs from the United Nations Pollution Control Agency. So examples of emerging contaminants, we have all pharmaceutical, current emerging contaminant components are pharmaceutical and personal care products, antibiotics, drugs, hormones, anti-cancer drug, veterinary drugs, all kind of drugs, personal care products, your soap, your lotions, sprays, etc., etc. All those materials under this personal care use and pharmaceutical component are part of this PPCPs. We call this PPCPs. Next biggest interest is on endocrine disruptors. A lot of research is going on endocrine disruptors. We will see in the subsequent slide about what are each of these pharmaceutical or endocrine receptors, etc. The other new emerging contaminants is microplastic pollutants and nanomaterials, so on so. So going to the source of emerging pollutants, all kind of anthropogenic activity, right from your household activity, industrial outputs, hospital runoff, whether it's waste from originating from agriculture sector or animal husbandry, all can give rise to emerging contaminants. So these emerging contaminants, if it is treated, it gets into the waste disposal system and it ends in your aquatic stream. As you know that all kind of pollution, wherever it may be, whether it's disposed on land or in the air, water acts as a sink. So eventually everything lands up in your aquatic system, whether it's a freshwater or a marine system, it's a groundwater or surface water. So that is a very important point to note, because emerging pollutant at last it enters into the aquatic streams, and we may be seeing some points where the actual quantity of the pollutant emerging we call as ECs or EPs will be very, very minute, or in only in we can say parts per billion, but in spite of the concentration, it leads to bioaccumulation and causes various problems to aquatic flora and fauna. So that is the aspect of EC on environment. This slide is about the health impacts on humans. It can cause so many issues, right from your endocrine hormone disruptions leading to thyroid gland problem, hormonal imbalance, fertility problem, cancers. It can cause reproductive problem. It can cause teratogenic problems. There are so many issues, all kind of health impacts it can cause. So it's very important to identify the emerging contaminants and to handle them. So what are endocrine receptors? Endocrine receptors are mimics or hormonal mimics, where it can affect cause impact similar to some hormones may not be needed for certain animals. Because of this endocrine receptor present in the animals, it can cross that kind of hormonal response in that animal. It can stimulate or retard growth. It can affect the reproductive system. So even at a very, very low concentration, very low exposure, it can cause the hormonal effect mimicking what's happening in humans. So different types of endocrine receptors, we have bisphenol A, DDT, estrogenic substances, phthalates, we call this plasticizers, which are used in toys, fire retardants, etc. So next component is your personal care products, pharmaceutical and personal care products. It's a very diverse group of chemical enclosing all kind of drugs, diagnostic agents, like what is used in X-ray contrast media, various consumer chemicals, and excipients, components, ingredients used in manufacturing your drugs, formulations, etc. So why particular PPC is more significant is that the persistence of this emerging contaminants, it has been recorded over the past 2 decades, 20, 30 years. Even though natural degradation happens, even though natural degradation happens, settling happens, even the concentration, what is let off is less, it accumulates, it becomes persistent over the year, get into the food web, food chain, and it causes numerous problems to the aquatic fauna and flora. So this slide shows you the various sources of the PCPCs. It's not very clear image, but like you can see from the top hand side, 1 year it's household activity, 2 is hospital waste disposal, 3 is private septic tanks and sewage collection areas, 5 is a direct disposal of PCB into the aquatic streams, 8 is aquaculture, 9 is your pesticides into the farming areas, and 10 is your fate of the PCBs. As I said in the previous slides, the entire different origins of ECs, emerging contaminants, irrespective of whether it's being PCPCs, ends up in aquatic environment. So what are the concerns, ramification, or the problems is that, see, drugs, when human consumes, it does not a concern because it's based upon the body weight. It's a very small dose and it causes a therapeutic gain. But the aquatic fauna or flora, they don't need this particular emerging contaminants or PCPCs. So the exposure causes varieties of problem, and not everything is actually studied deeply. It's very, very difficult to ascertain the actual impact of each of the components to different flora and fauna in the aquatic system. So aquatic organism can cause can suffer continual exposure, even though the concentration is very, very less. Due to cumulative and synergetic exposure, they cause a lot of problems to these organisms. So bioconcentration is a different terminology for bio magnification. You have all these ECs getting up into the microorganisms, macroorganisms in the form of aquatic flora and fauna. irrespective of the nature of the ECs, whether it's polar or non-polar. As you can say, certain drugs, despite the low lipid solubility, are detected in aquatic tissues. So the concentration is much in higher number. Like for example, estrogen, it's 60,000 more. What is actually should be present? It's a traces. So your gemcitabine is it's a lipid control drug for human, which is found in 113 times in fish tissues. Diclofenac, it's an anti-inflammatory drug. It's again found in various fish organs, 2,700 times more than the permissible limit. And your flucosectin is an antidepressant. It's found in muscles, liver, brains of fishes. So various studies shows that biomagnification has occurred in aquatic plants and animals. So next component of your ECs are microplastics. Microplastics is now recently into focus of much interest to scientists across the world. We hear about news about the occurrence of plastic-like materials in fish and other sea animals. So this is of very interest, and there is a very big need to identify, characterize, and also to mitigate this microplastic menace. So microplastic can be classified into 4 categories based on their sizes. It can occur from your nanoparticle size, 1 micrometer to a few millimeters. So can be classified based on their size into macro, meso, micro, and mini micro, or it can be classified based on the type of occurrence. It can be in a fibrous form, it can occur in a pellet form, or it can be a fragment or a foam or a film. So the other way of classifying your microplastic is primary and secondary microplastic. Primary microplastic, as it shows very clearly, it directly enters into the marine environment, which is actual size. Secondary microplastics arise from the mother or the original plastic material, large plastic material, and it get degraded due to natural activity, the sunlight and the marine salt. It leaches the plastics and it get disintegrated, break down into smaller microplastics. So these are the ways of classifying your microplastics. This is a very interesting slide as a research point of view, because as you can see, naturally in the aquatic environment, especially in marine ecosystem, biodegradation naturally happens, photo-degradation also naturally happens, which itself take care of the immanence, but at the same time it itself giving rise to new problems. Biodegradation of larger plastic makes much more microplastics. Same goes for your photo-degradation of larger fragments of plastic, causing more microplastics. So a problem itself, a solution itself is a problem. So so it's a very interesting paradigm where we are in a quandary whether which way to go about. So as you can see, the below the slide, some biodegradable plastics have been made from starches. These are the alternatives. So solutions to this thing, but still conventional plastic is used in large volumes, and microplastic is becoming a very real life hazard. The next class of or component of your ECs are your nanoparticles. It's a new emerging field. Nanotechnology is a new emerging field. Past 2-3 decades, a lot of nanomaterials and nanoparticles have come. A lot of research is going on application of nanoparticles, but very few less studies have been made. Very few data available on the hazard of nanoparticle and nanomaterial, but the data is quite sufficient enough to include them as an emerging contaminant. Even nanoparticle is used in personal care product like sunscreen, lotion, etc. So they get into the pore skin and they enter the body. If it is they can enter the into the lungs. It can cause respiratory problems. So as you can see, it can penetrate into human body if inhaled, and it can pass into the bloodstream, and it can travel to all the organs in the human body. Some nanomaterial like your multi-wall carbon nanotubes has been reported to cause asbestos like effects, pneumoconiosis, and so many other mimicking diseases. As I said earlier, there is a huge lack of data available to ascertain the exact hazard of each of the nanoparticles or the different types of nanoparticles. But still, it's quite enough to classify them as emerging contaminants. We can judge to a certain extent the feasibility of what this nanoparticle can cause an hazard based on their bulk material or the starter material. But interestingly, a lot of nanoparticle have shown that the property is contrary to the bulk material. So it's carefully we have to assess. It's one pointer. Maybe certain nanomaterials and nanoparticles can be evaluated. The health hazard can be evaluated based on their bulk materials, but it not be applied for all the nanoparticles. So the toxicology of the nanoparticle definitely depends upon the size, the surface charge, the age of the nanoparticle, the type of the nanoparticle. So there is a need for a particular regulations, need for proper guidance to study nanoparticle, to assess the hazard of nanoparticles. So nanoparticle effort, what are the concerns? The possible concern is that we need to be aware of nanoparticle. We need to be very careful to handle the nanoparticle. That's the need of the hour. If at all if we get exposed, it can cause cancers, it can cause pulmonary diseases, it can cause cardiovascular diseases. It can, as I said in the previous slide, can travel through the bloodstream to all the organs. And interestingly, that's a point I need to tell you. We have something called as BBB, blood-brain barrier. So brain is protected, and generally, very rarely, a microbe can breach this wall. But it's not a scenario for nanoparticles. Nanoparticle can travel and enter into the brain. So that's again something to worry about. So it can cause DNA repair, DNA mutation, cancers, and other organ damages like liver. So now we have seen the hazards and the types of emerging contamination, all this thing. And now I'm going to why we need to do research. What is the actual focus of interest of research happening? So this is just a table calculated based upon software to see how much of published data is available in the Scopus database and which are the leading country who are doing research on emerging contaminants to mitigate, to remediate, to degrade, whatever, to handle the emerging contaminants. And India is on the top 4 highly published papers. And if you think about the various keywords which can be used associated with emerging and how to degrade, biodegradation, all this thing, we can see different scenarios. But a lot of research interest is there on this emerging contaminants, and a lot of quality research outcome coming out. So this is just a network visualization map for coinage has been done. If you just search the Scopus database or any or even your SCI database, combining your emerging contamination with biotechnology, handling biodegradation, all these things. So this is just a map. So to give you an idea about the interest focus of interest, how so many different fields of biotechnology can combine and they can handle these emerging contaminants. Obviously, when you want to talk about the biological means of management of emerging contaminants, the most predominant route is your bioremediation. So you know about the definition of biotechnology. So any biological means you use to to improve the quality of life, it's biotechnology. So bioremediation is one avenue where you can apply and you can mitigate this emerging contaminant issue. The next is your phytoremediation. I don't want to go into actually how this bioremediation occurs or how this phytoremediation occurs. Phytoremediation, if you, as an environmental, if you want to talk, I'm not much interested because the phyt plants accumulate very fast all kind of pollutions, and it will die after some time, and then there is a necessity for to dispose this big mass of biomass, then probably recovery, all these things not economically viable. So phytoremediation is very interesting, but a lot of improvement is needed. We need to identify very specific plant types which can be not a commercial crop and still can be used for this phytoremediation, especially for this emerging contaminants. So when you want to talk about biotechnology, microorganism can actually play a better role than plants. So this is how phytoremediation can various avenues in phytoremediation where how we can use and we can handle this emerging contaminants. So coming to the microorganisms, obviously we have these three different classes: bacteria, fungi, and algae. And bacteria, very less studies have been done because bacteria has its own limitation, very small organelle. Either it has to produce a single enzyme to take care of a particular pollutants. So the advantage lies with fungi and algae. So it's a macromolecule, macroorganism, algae, and that can be easily exploited in a sustainable approach than using a bacteria. So we know about various cases data available, like microalgae, chlorella, or the conventional fungi in the form of aspergillus or penicillium, or so many bacteria have been used to uptake these ECs. When you want to go for talk about the microbial degradation or microbial handling of emerging contaminants, so there are only two paths: either biodegradation or adsorption. So you have there are certain components, but it's very less quotient where biotransformation also occurs, but it's a completely different dimension. It's a very interesting topic. We can even have a very good talk on that biotransformation as such. What are the different emerging contamination? It can be like a biocatalysis, all these things. So it will be too deviated. So I'll just stick to these two things: biodegradation or removal through sorption. So in a single slide, I want to show you the various parameters, limitations, and various treatment methods which are available pertaining only to the microbial system, how it can be used, HRT or the temperature, the pH, substratum, and all these things. So just a nutshell, a single slide, single picture I'm just putting. So for handling of ECS, a lot of studies have been done. Very limited studies have been done compared to fungi and algae. And one example is Aurelio Romero in 2022. Recently, he has described about a new concept: the bacteria and any other organism. As I said, no, the bioaugmentation is you are enriching the particular bacteria or a microorganism to getting exposed to a particular pollutant, like a particular emerging contaminant, and due to this exposure, it empowers the particular bacteria or a microorganism to handle this emerging contaminants in a better way. Okay, so with this system, they were able to eliminate about 99% of the those materials they had tested in their system within 25 days. So this is what they did. They have went for this new BPS accumulation system, where they have exposed the bacterial cultures to the pharmaceutical antibiotics and the chemicals like diclofenac and ibuprofen. And because of this bioaugmentation, the exposed bacteria have been empowered to handle this EC much more. So they called us the system. They're this word called bioaugmentation. And you have a unique group of bacterial community which can be harnessed in a larger scale for handling these emerging contaminants. So this is available in literature about recent literature about various emerging pollutants and emerging contaminants and various bacterial sources which can be used or which has been used, and these are the various percentages of degradation achieved by the various bacteria. So this is how fungi can interact with a particular EC. Here the example is taken as diclofenac. Usually it is either through ion channels available on the cell surface, or it can be via complexation. Here a diclofenac is used as an example, how this is taken up inside the cell and how the fungal handle and how they degrade this particular emerging contaminant. So there are various mechanisms how the fungi will attack the emerging contaminant. Essentially, the starts with the emerging contaminants getting attached to the cell wall. It is somehow similar to what a bacteria does also. So the EP get adsorbed to the cell wall, and it gets into the system. Sometimes the fungi produces special enzymes. We call that as surfactants or biosurfactants. So this or it can be reductase enzymes. Various reductase enzymes are known to be produced by the fungi, like lactases, lignin peroxidases, lysozymes, so many things which can effectively attack these emerging contaminants. And fungi is a better media to handle the emerging contaminant than the bacteria. So a typical example is this Dalika et al. In 2021. They have studied on the pharmaceutical emerging contaminants, and they have chosen their own fungal consortium like your Aspergillus and Trametus, and they were able to use the fluidized bed reactor to efficiently remove this emerging contaminant. Up to 90%, they were successfully able to eliminate. So the pharmaceutical components studied here were ibuprofen, ketoprofen, metoprolol. So they are successfully claiming that their fungal isolates were able to handle this emerging contaminant. So these are the various other examples available in literature. As you can see in the left-hand side of the table, you have various emerging contaminants, and the middle column goes for this various reported fungi, which able to successfully, from average, you can see 70 to 90 to some compounds like triclosan have been 100% degraded. So fungi are a better agent than compared to bacteria, and even better agent is your algae. So when you want to explore your emerging contaminant, best tool, you can say very easily algae are the best tailor-made natural gift to handle your emerging contaminants. So these emerging contaminants enter the algal cell wall, and it undergoes your biomineralization. Unlike your bacteria and fungi, you have enzymatic conversion of this thing, but here kind of biosorption occurs and biodegradation occurs in the form of biomineralization. So this is the most efficient this thing. Algae also degrade this emerging contamination with the help of photo-degradation. So this is in a nutshell about how most of the algae, whether it's a macro or a micro algae, how they handle this emerging pollutants or emerging contaminants. So these are the various examples available in literature. You have the left-hand side the emerging pollutants or emerging contaminants, various algal bodies. Most of them are microalgae, marine algae, and you can see consistently 70 to 19% achievable efficiency was achieved for most of the emerging pollutants. So examples, to give you specific examples, Matamoros, they were able to handle the emerging contaminants, which here they say is pharmaceutical wastewater, with the help of the microalgae chlorella in an aerated batch reactors, and they were able to completely degrade. 99% efficiency was achieved within just 10 days. So they say their system is microalgae is better agent to handle this emerging contaminants. And Roas et al. 2022 evaluated their own system. where they had a integral microalgae based domestic wastewater treatment coupled with your anionic reactors and aerobic photoreactors, and they were able to they took around 16 to 17 components, various emerging contaminants, right from your penicillin to triclosan, and they were able to achieve 80% degradation to most of the emerging contaminants what they had in their study. So these are the various examples where I can give say that a microalgae or algal system is a better one to handle emerging contaminants. So this is a specific example how a particular microalgae or an algae can degrade antibiotic. This is a symbiont example. This is a combination of microalgae with bacteria, bacterial biofilm with microalgae, where this can be also exploited to handle your emerging contaminants. So instead of stopping only with conventional technologies or biotechnology, the various literature says and shows that hybrid processes are a better way to handle your emerging contaminants. Instead of going for either conventional method or a single system, it's always effective to go for a multiple systems. So hybrid systems examples, combination of your RO membrane or catalytic oxidation or UV based oxidation, all these things you need to couple with your biotechnology concepts, taking into consideration including algal system or a fungal system or a bacterial system. So if you go for hybrid systems, your efficiency will be improved with lesser HRT. We call this hydraulic retention time. When you want to go to industry, most of the research you can see that it will be like a lab scale. So the volume will be very less. When you scale up, the efficiency will usually drop. So industrial point of view, HRT, the hydraulic retention time is very important. That whatever waste it has to be handled within a single day, it cannot accumulate. So hybrid system is the best way to go forward to handle your emerging contaminants. So this is the future prospects, future scope. Emerging research in emerging contamination mitigation is your nanobiotechnology. You can debate that, sir, you're saying nanomaterials, nanoparticles, they themselves are emerging contaminant, how nanobiotechnology will be. We need to go for a technology where the problem itself will be a solution. So in that context, nanobiotechnology has a better say. Like nano itself, when it promises a lot of surface area, so you have too much of space to exploit. So that is a better system to handle this thing. So this is a rising upcoming research areas apart from your hybrid systems, where nanobiotechnology can also effectively handle your emerging contaminants. As you can see from the table, like so many emerging contaminants has been reduced 70 to 100% with the nanobiotechnology system. So this second slide is a continuation of the previous slide. So I have finished my talk. If you have any questions, you are welcome.
Yeah, thank you, sir. Now I request the participants to discuss with our dignitary. If you have any doubts or clarification you want to discuss, like you can put your question in the chat box. So the question is about, someone has asked about phytoremediation.
Yeah. I already discussed about like from soil. Can the question be clear, please? See, phytoremediation for any pollutant handling, I am not for it. It's not actually helpful. As my experience goes with phytoremediation, So as I told you, now if you're using phytoremediation, you may able to successfully remove large scale of a pollutant, but the whatever plant you're using, it will die after some time, and you need to dispose the plant. Hazardous now, the hazardous substance has got into this plant. How will you dispose this large biomass? So that is a bigger issue. We are not, see, solution should be a solution. Solution cannot be a problem.
Only one question. Sir, do you have any idea about the role of specific actinomycetes in the field of microplastic degradation?
Microplastic degradation. You want to go for actinomycetes?
Yes, so the question was like that.
Degradation, yeah, anything is possible. Actinomycetes, see, degradation of anything, whether it's an emerging contaminant or normal pollutant through bacteria, or actinomycetes, a bridge between bacteria and the fungi. So even though it is an ancient bacteria, we prime it, it has to produce some kind of enzyme to handle the particular thing. So when you want to go see literature, polyhydroxyalkanoates have been degraded by lipases. So if that particular actinomycetes should have the capacity to, or should have the plasmids to handle this thing. Otherwise, it will be very difficult. Polyhydroxyalkanoates is not within the topic. Why somebody is interested in polyhydroxyalkanoates?
Okay, the next question is.
Yeah, you see, yeah, all the systems can help this, including phytoremediation. But the problem is handling phytoremediation is very difficult. Like, you need large resources. You need to have hectares of land, and you need to dump that. We have a constraint for land resources. Then you have to plant hectares of plants. Then it will definitely, it's one work. Initially, it will work wonder because it will uptake compared to bacteria or any other microorganism. The volume of handling will be very, very promising. But the problem will be it will die, and we need to dispose, again disposing. So it is just shifting a particular pollution from one side to the other side. Yeah, you may argue that there can be some biotransformation goes on, but I don't come across a system where plant has uptaken and completely transformed all kind of this thing. See, even if you take a heavy metal, it will accumulate. So you have a very concentrated form of heavy metal in that particular plant. What are you going to do with that? So that's the question. We can use plant for even uranium, radionuclei, and any other metal, but the metal will accumulate in the plant and the plant will die. And what are you going to do with the dead plant biomass? Yeah, it'll again you need to dispose. No, see, initially you require the hectares of land first of all to plant it, whether it's a reed or it's really a terrestrial plant like big trees or shrubs. Then you need to expose that any pollutant, and it will definitely uptake very fast. The hydrofiltration time will be very wonderful. And again, there is another problem. You need to grow the crop. It will take its time also. So if you think about it, you can see so many issues with phytoremediation. Even as an editor, I don't encourage phytoremediation papers. Maybe you may call it I'm a bit biased, but as far as my limited exposure goes, so many papers I've seen, it is not a solution. It's just a switching problem from one end to other.
Sir, there is a question from my side.
Yes.
One question. As you said about carbon nanoparticles as one of the pollutant, as an emerging pollutant.
Yes.
But carbon nanotubes, and say whether it is a single wall or multi-wall, it's currently nowadays widely they were used in the water treatment process itself.
Yes. It is.
So could you please correlate and tell me how it will be the again emerging pollutant and controversially it has been used?
What is a pollution? We need to go with the basis. What is a pollution? There is a saying called one man's food is other man's poison. Oxygen is a pollutant to anaerobes. Am I right?
Yes.
So yeah, so it causes health hazard. Nanomaterial causes health hazard. See, every coin has two sides. There is an advantage and disadvantages. Okay, there are a lot of research coming where showing hazards of nanomaterials. It's not that we should completely stop. There are a lot of pluses with nanomaterial. I told you, like infinite surface area. So you can, it's a very easy system to, simple system where you have exponential surface area, so that gives a lot of promise to handle various pollutants, including emerging contaminants. So whether it's a CNT or any nanoparticle, it's a wonderful tool to, you can even go for composite materials from the nanoparticles. You can go for nanomembranes. There are so many aspects of nano research, very interesting, very promising. But at the same time, it is also causing problems. Like any system we have, it will cause some health hazard, and the data is showing that there is causing health hazard, and there is limitation. We don't know the complete picture. That is the problem with emerging contaminants. We don't know what will happen. It is causing problem. It has a potential to cause a problem. That is the terminology of emerging contaminants. It may cause a problem. Like microplastics, we don't know. We don't know. We say that microplastics can cause cancer. It is actually clogging into soil. It is actually getting into so many fish and other aquatic animals. It is actually causing death of a lot of marine animals. So that's the thing. So emerging contaminants is, we don't know the actual quotient of the. It's very difficult to ascertain. See, in a system, if we have say drug is there, drug can be studied whether this drug can cause a problem. Okay, but we cannot say about emerging contaminants because we don't know how many quantum of exposure. There are millions of species of microorganisms there. There are hundreds and thousands of other organisms are there. We don't know. We still, each day we find in newspaper new frog, new snake has been discovered and all this thing. So we don't know the quantum of this pollutant is going to cause various. See, a very ppb concentration of a particular hormone will be beneficial to human beings, but causes so many adverse effects in many aquatic plants and animals. So we cannot actually imagine the extent of the hazard it can cause. That's a problem. That's a problem of emerging countries. That is why so many studies has to be done. It's a very upcoming field. It's a lot of potential, lot of interest, and it will be very useful for all students, whether they are graduates or postgraduates, they are research scholars, or even assistant professors. A lot of fundings are available. You can write interesting proposal. All these avenues you yourself, the person who asked the question, can even give a proposal how carbon nanotube can be a problem or how it can be addressed. That can be a research proposal.
Yeah, thank you, sir. Well explained. Thank you. Thanks a lot, sir.
Thank you. Welcome.
Any other questions from the participants? If there is no question, we will move on to the conclusion. And now I would like to invite my colleague, Dr. Priya, Department of Biotechnology, to give the vote of thanks. Ma'am, over to you.
So can I leave?
Good evening to one and all present here for this 5th day of 1 week professional development program on avenues of biotechnology. On behalf of Department of Biotechnology, SRM Institute of Science and Technology, Ramapuram, I want to extend my warmful thanks for our today's guest speaker, Dr. J. Arvind Sir, Adjunct Professor, Department of Biotechnology, Savita Institute of Medical and Technical Science, Chennai, for accepting our invitation and his valuable and interesting talk on sustainable approach for handling emerging contaminants through biotechnology. Yes, sir, today we got a clear idea on current unsustainable situation, terminology of emerging contaminants, sources of emerging pollutants, pharmaceutical and personal care products, microplastic, and phytoremediation. Hope our participants also enriched knowledge with these emerging contaminants and their handling methods. On behalf of our organizing committee, I want to thank mainly our participants, especially the faculties from many schools and colleges for your valuable presence on this today's session and making it a successful one. We are expecting the same support for upcoming sessions also. Once again, I thank you, sir, and thank you all.
Thank you, sir, and thank you to the participants. The feedback link is available in the chat box. I request the participants to kindly fill the feedback link. Thank you one and all. Arvind sir, thank you, sir. Thank you so much, sir.
Yes, sir.
So thank you so much for accepting.
Somebody asked me how
invitation.
to reach me, so I don't know whether this is a correct platform to post, but I have given my mail ID and phone number.
No problem. So if it is, we will get it back to you.
Yeah.
Okay. So thank you so much for accepting our invitation and for your time.
It's my pleasure to talk to you. Thank you.
Thank you. Thank you, sir.